Okay, now let's talk about the liver. So as I said earlier, histologically, the liver has a lot of parts. No, some are imagined and some are seen histologically. So as a review, the liver is actually, let's talk about the blood flow within the liver. No, so for the liver, actually, blood flow is from the aorta, and then it will go to the different parts of the GIT system, and then this blood will go to the portal vein into the liver, wherein the nutrients that they absorb as well as possible pathogenic things that need to be detoxified will be processed in the liver before finally going out of the liver and into the hepatic veins and into the caudal vena cava. And then for the oxygenated blood of the liver, it actually comes from the hepatic artery. Okay? So for the venous system of the liver, it has the portal vein and branches of it that brings the oxygenated blood nutrients to be metabolized as well as possible pathogenic things that need to be detoxified from the GIT system. After processing it, it will be put into the main circulation. And then the oxygenated blood comes from the hepatic artery branch of your aorta. Okay? So it looks like this. You would have this part, the portal vein, here, and then the hepatic veins actually a little bit more outside the actual liver. That's why histologically it would be the portal veins and the branches of it that you would see in the portal triad. Okay? So here you can see portal vein, the hepatic artery, as well as the bile ducts that will be seen in the portal triads histologically. Okay? So another schematic diagram here, you would see the liver lobules. Okay? So these liver lobules, they would have centrally located central vein. Okay? So from the edges here, that is where your portal vein earlier, this one, and your bile duct, sorry, this one is located, as well as your hepatic artery. So this one. So the flow of blood is actually from the edges of this liver lobule or classical lobule. So from the edge, so these three, the portal triad, are located here, 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 and the blood flow goes into the central vein. While the production of bile produced by your hepatocytes, goes in the opposite way from the central area into the portal triads. Okay? Again, we have our central vein and then the portal areas. And then multiple versions of these would compose the whole classical lobule. Okay? So you would have your hepatic cords and your sinuses, or the areas where blood is. Okay? Histologically, it looks like this. So what we have to do is look for the central veins, okay? So central veins usually have this direct connection with the sinusoids, okay? And then on the edges of the central veins, you would have your portal areas, wherein it contains the structures that we talked about earlier. The bile duct, which is lined with simple cuboidal epithelium, small artery or arteriole maybe, we can call it as an arteriole or your hepatic artery, and then branches of your portal vein. Okay, so this whole area is termed as your portal area. Again, this is classic lobule, central vein. Blood flows from here to here. Bile flows from these hepatocytes to the bile duct. Okay? Again, Here you have your hepatic cords or row of hepatocytes. And here you would have your portal area, portal triad, because there's one, two, three structures within it. Okay? Bile duct, hepatic artery, portal vein. And it corresponds to this schematic diagram. For the hepatocytes are darkly staining because it's working a lot. It contains a lot of Golgi apparatuses for the production of secretions, and it has mitochondria, which needs energy for the metabolism and absorption of stuff. So that's why hepatocytes are darkly staining. Some hepatocytes would have single nuclei, while some would have multiple nuclei. And here, as told earlier, because of the abundance of organelles, the cytoplasm is granular and darkly staining. And here you can see that these are your cords, and in between them, you would be your hepatic sinuses. Earlier, we talked about how these sinuses is actually a type of sinusoidal capillaries, right? And one of our clues is that we can see RBCs within it. Okay? How about these cells? What are these irregular or stellate-shaped cells? These are actually your Kupffer cells. Your Kupffer cells are just another term for the macrophages found in your hepatocyte. Okay? So these are your Kupffer cells. Again, you have your Kupffer cells lining the blood sinusoids. And actually, aside from the blood sinusoids, we also have this structure which we call the space of this, which is basically a very thin space in between your hepatic cords and the hepatic sinusoids. But actually, most of the time, this space is not easily seen or already artifactually removed from the sections. Okay? Again, this is the portal triad. Okay, now let's go into the lobules that we have been talking about earlier. First, as I said earlier, the histological lobule, which is seen under the microscope and divided by connective tissue, is the hepatic lobule. Okay? So the hepatic lobule basically tells the story of the blood flow, which is actually from the hepatic artery going into the central vein. So hepatic artery and the portal veins actually go into the central vein, okay? So that it can be drained into the vena cava finally, okay? Into the hepatic vein and finally into the vena cava. Then we have our portal lobule, which basically tells the story of the bile flow, because the central part of your portal lobule is your portal triad, because bile produced by the hepatic cells or hepatocytes in the hepatic cords will actually flow from the center of the hepatic lobule into the portal triad. So this is your portal lobule. Again, the edges or the corners are actually your central veins, okay? With the portal triad, that's why it's called portal lobule at the center. Next, we have our hepatic sinus, which actually tells the story of the metabolic activity gradient and the oxygenation levels of the cells within the liver, okay? So here you can see that it's actually made up of two central veins and the cells in between them. So two central veins and then portal triads. Okay? So there are actually different zones here, with zone 1 being nearest your portal triads, your arteries, and then zone 2 in the middle, and zone 3 being nearest your central veins. So, again, as a review, this area, your portal triad, are the ones located here. So the bile, Direction is from the central vein to the edge. So bile direction, central vein to the edge. And then for the hepatic sinus, as I said, there are different zones because here you can see um, the portal vessels and then the central vein. So it depends on what activity or oxygenation level you are trying to analyze, no? So in the zone 1, this area has a lot of gluconeogenesis, beta oxygenation, and is receiving more nutrients and oxygen because it is near your hepatic artery, right? Also, because of that, it is also very susceptible to infection and cirrhosis because the blood supply enters this zone first. While the central vein area near it, zone 3, would have less oxygen, less nutrients, but it would have more glycolysis and lipogenesis and other signaling. And then this area is also susceptible for fibrosis, parasite infection, as well as alcohol toxicity. So depending on the type of injury, you will see that they have a tendency to develop either near the central vein or near the portal areas. Okay? So here is your classic liver lobule with the central vein at the center and then the portal triads at the edges. And here you have a portal lobule with the central veins at the edge and the portal triad at the center. And here you have the imaginary, well, the portal lobule is imaginary as well, but imaginary um, portal asinus, liver asinus, or hepatic asinus, wherein you would have the different zones that is going nearer the central vein and farther from the central vein or nearer the portal vein or farther from the portal vein or sorry, portal triad. Okay? So again, the different zones. Zone 1, 2, and 3. This is another look into the three lobules. Classic lobule, hepatic lobule, aka hepatic lobule. And then here we can have our portal lobule, central vein, central vein, central vein, central vein, central vein, central vein, portal triad, portal triad. Okay, then we have our hepatic asinus, which can be seen here, central vein, portal triad. Nearest the fresh oxygenated blood supply and nutrients, zone 1. Zone 2 in the middle, zone 3, least supply of nutrients. Zone 3, usually for toxicities, parasites. Zone 1, inflammation, and um, infiltration of bacterial infection. And zone 3 is also susceptible for um, not being able to receive oxygen because it is farthest from the arteries. Okay, so some example of histological or histopathological slides of the liver. Here you can see the central veins with necrosis or dead hepatocytes. So you can see it's already coagulated, while the hepatocytes near your portal areas, hepatic artery, are still managed to survive during um, more earlier stages of liver inflammation or liver disease. Okay, here is a zoomed out version. You can see here that areas near the central vein look pinkish and that you cannot see the specific dots of hepatocyte nuclei. It means they're already dead. While, so this is zone 1, while the zone 2 and 3 The hepatocytes are still alive, but still weird looking because you can see the white, bubbly, vacuolated appearance, which is some form of, which is called steatosis or the infiltration of lipid. So this may be seen in livers of people who are alcoholic, no? Then another version of this, central vein, portal triad, zone 1, 2, 3. 2 and 3, you have steatosis. And then here you can see in the portal area, there are inflammatory cell infiltrations. Zoom in version, portal area, since it is near the hepatic artery, it and portal veins, it can bring infections taken from the other segments of the intestine. And then remaining hepatocytes in zone 2 or 3 near the central vein can show lipid accumulation as a form of injury. Okay, so again, all of these show that the value of knowing the different zones, because depending on the zone that is affected or that is dead from injury, the histopathologist can have an idea of the cause of liver injury. Okay, next we have your gallbladder. So for the gallbladder, it looks like a segment of your GIT tract because it also has a simple columnar epithelium in the mucosa. It also has a lamina propria connected with your submucosa. And in some species, this area would actually contain glands. And then it would have a not as extensive muscle layer. And then it can have an adventitia or serosal layer. And here you can see that there's a lot of mucosal folds, no? Which kind of looks like villi. So you have to really view the slides in order to differentiate the gallbladder from, let's say, the small intestine. But I would say it's usually not the small intestine because there are not much um, goblet cells in the lining epithelium. And then the muscularis layer does not have the inner circular nor the outer longitudinal layers. Okay, what else? Here you see extensive um, mucosal foldings. And if we zoom in more, zoom out, here you can see that it's directly attached to liver parenchyma. So you would say that this area would have a adventitia outer layer, and this area which is not in contact with the liver would have this serosal layer. Okay? If we zoom in the simple columnar epithelium of your gallbladder, we can also identify light cells, which constitutes majority of the columnar cells, and then you would have your dark cells. Okay, so these light cells and dark cells apparently play different roles in the gallbladder. So majority of the light cells would be the ones which would have roles in concentrating the and absorbing excess water and concentration of the bile, while the dark cells would be, since it's dark, it means it's more active, it contains more organelles which produce something. So references say that these dark cells instead of absorbing excess water in the bile being stored here in the gallbladder, it actually produces some bicarbonates or mucus secretions which would um, temper or lower the acidity, excess acidity of digestive juices. Okay? So here is a liver from which species? If we look closely, the RBCs are nucleated. So this is actually a slide. I just want to show a species difference. avian liver. And you can see the avian liver is basically the same structures. Here, we have a close-up of a central vein, but since it is from the avian species, the RBCs have nuclei. And still, you have your hepatocytes, hepatic cords, and hepatic sinuses. And still, you would have your portal triads. In this specific photo, I think there's a lot of infiltration as well around the portal triads. Okay? A key difference in avian species is that the connective tissue surrounding the classical lobule is not as extensive or well developed. This holds true with humans as well, okay? The classical lobules is very difficult to ascertain, okay, or to see. While in the pigs and camels, this is opposite, no? The connective tissue septa in between the hepatic or classical lobules is very thick and well defined, okay? So here are other photos of the human liver. Just like the avian liver, the lobules are not well defined, so you have to really differentiate the portal triads from the central veins in order to see where one lobule starts or and one lobule ends. Okay, so in the lab, please view the pig liver first, so you would have a clear delineation of the classic lobules, and then from there you can easily trace where the portal lobule and portal acinus are as well. Okay, so attendance. Okay, and for those who are still listening and reach this point, please do not tell this to your other classmates. I am offering a bonus for you. So please send an email to me with a picture of your favorite animal. It may be your pet or a random photo in the internet. Just put in the subject VETA 106 cute animal bonus and just send it to me anytime within the semester. Again, please keep this a secret, and it's a bonus for you guys who are still viewing this video, even if it's already around 22 minutes. Okay?